This is Module 5 on translation. In this video, I'll describe some of the general features common to translation, including some features of the tRNAs that are involved, how tRNAs are linked to the appropriate amino acid. I'll describe the structure and function of the ribosome, and then briefly touch on the 3 different stages of translation: initiation, elongation, and termination. Translation of a messenger RNA, so the code embedded within a messenger RNA, is translated into proteins by an enzyme called the ribosome. The ribosome is a multi-subunit enzyme. So there's a small ribosomal subunit pictured here on the bottom and a large ribosomal subunit pictured here at the top. And all ribosomes have embedded within it small sections of RNA called ribosomal RNA or rRNA. A ribosome will recognize AUG as the start codon for all proteins. And so all proteins begin with AUG and therefore will begin with a methionine, since AUG encodes for methionine. The ribosome will start creating a polypeptide chain, starting first with the N terminus, linking peptide bonds along the chain and ending with the C terminus. And the ribosome reads the messenger RNA in a 5' to 3' direction, starting with the AUG start codon and ending with one of the 3 stop codons. Let's take a look at the tRNA players. So while the ribosome is responsible for creating the peptide bonds between 2 neighboring amino acids, the tRNA is responsible for bringing the correct amino acid to the ribosome in the first place and helping to decode the mRNA. tRNAs, we've mentioned before, have this cloverleaf structure where they have the anticodon, which is complementary and antiparallel to the codon in the RNA. And it also contains an amino acid coupling site where the appropriate amino acid is linked to this tail, the amino acid that correlates to this code. The enzyme that charges tRNAs or links an amino acid to it is called the aminoacyl tRNA synthetase. And it has 2 active sites, one where the tRNA binds and the other where the appropriate amino acid binds. And so every tRNA has a unique shape that will fit into a unique tRNA synthetase. And the tRNA synthetase also has this active site is unique to the amino acid that fits in it. And so because these 2 fit in so perfectly, only some amino acids, some tRNAs can fit in this site, and only one type of amino acid can fit in this site. So the specificity between amino acid and its tRNA is determined by this enzyme. There are 20 different aminoacyl tRNA synthetases and about 30 to 50 different types of tRNAs. And so there's a little bit of degeneracy in the tRNA synthase molecule as well. So one of these enzymes is capable of fitting maybe a few tRNAs, but only one amino acid. This enzyme will link the amino acid to the amino acid coupling site, the acceptor arm. And so that's the 5' end of the folded up tRNA. And it will be the opposite end of the anticodon on the tRNA. Here's this pictured again. Here's the anticodon on the tRNA, and instead of flattening it out into that cloverleaf structure, we're going to fill it in a little bit, so a space filling model. Here's the acceptor arm, and on the acceptor arm, there's this CCA motif, where you see CCA in nucleotides appearing in this order on the 3' end on the acceptor stem. Okay, so when we take a closer look, how is the amino acid being linked to the tRNA? The carboxyl group of the amino acid is linked covalently to the hydroxyl group on the 2' or 3' carbon atom of this nucleotide, of the adenine, the final nucleotide on the tRNA end. So it can either link to the 3' OH or the 2' OH. This acceptance happens on the 3' end of the tRNA, specifically at this adenine group. And so that base is always adenine. And so the amino acid may be different, but it's always linked to its appropriate tRNA at this adenine site on the 3' acceptor arm. Certain positions on the tRNA molecules are recognized by the enzyme that is supposed to charge it. And so These are the positions in blue. These are the same in all tRNAs, and they can't be used by the enzyme to differentiate between different tRNAs. These all have to be the same because amino acids are all linked to tRNAs in the exact same way, in this way that I was just describing. However, the positions in red are very important in recognition of the tRNAs by one type of synthase. And so these will be unique depending on the type of synthase it's going to bind to. And the position in yellow is used by more than one synthetase. So that allows for a little bit of the degeneracy that we see, where a couple of different types of tRNAs will fit in here, but only one type of amino acid will fit in here. And so what allows one type of tRNA to fit in this site for that enzyme? It's the presence or absence of these red and yellow bases. All amino acids become attached to the appropriate tRNA in 2 steps. So here's our amino acid. In the first step, the amino acid reacts with ATP. And so this is our energy molecule, and this allows for AMP to be linked covalently to the amino acid. Once the tRNA comes in, there's an enzyme here that you're not seeing. So this is a synthase enzyme where the tRNA binds to the amino acid group, and then both the tRNA and the amino acid group are embedded within the enzyme, the synthetase enzyme. AMP is released, and a bond is reformed between the tRNA acceptor arm, so the adenine, and the amino acid that is appropriate. And so this arrow represents the enzyme, the tRNA aminoacyl synthetase. Translation, very much like transcription and DNA replication, consists of 3 major stages: initiation, elongation, and termination. We also have 2 kind of substages called tRNA charging, which is where the amino acids are linked to the appropriate tRNAs. We can't make a polypeptide chain unless we have tRNAs to help bring us some of those Legos that we need to create our chain, so amino acids. And then the last sort of substep is peptide release, where the peptide that is created by the ribosome is cleaved off from the ribosome and released into the cytoplasm. A number of additional proteins are required to make initiation, elongation, and termination proceed. And a lot of these are energetic processes requiring ATP, as is the case with tRNA charging, or GTP, as is the case with elongation. There are so many different components required for protein synthesis. These are specific for bacterial cells, but you don't have to memorize these or anything. I just wanted to give you an appreciation for how many players are involved in this process. So we already talked about tRNA charging, but in the process of initiation, you're going to need a template, something to start with. You're going to need that first tRNA bringing that first methionine that recognizes that first start codon. You're going to need a ribosome, both a large ribosomal subunit and a small ribosomal subunit. But then there's these initiation factors, these additional proteins that help to facilitate the rest of this process. This is true also for elongation. There's elongation factors that help to bind to the different stages, the RNA or the proteins, and help to facilitate the process of elongation. Still, in termination, there are release factors that bind to the ribosome and help to terminate translation. And so it's not just the ribosome, the tRNA, and the mRNA. We also need these initiation factors that help to facilitate the process. It's kind of like a transcription factor in transcription. What's really cool about an RNA molecule is that because RNA is translated in the cytoplasm where ribosomes are present in pretty great numbers. And so one single messenger RNA can have multiple ribosomes bound to it, all creating a protein. And so this is not meant to show you a sequential stepwise process of translation. This is one mRNA molecule being read by 1, 2, 3, 4 different ribosomes at different stages of growing the polypeptide chain. And so 4 ribosomes are translating a single mRNA molecule, and this allows for lots of protein product to be created from a single RNA molecule. Another feature that I want to describe is that translation and transcription are coupled in prokaryotes, meaning they happen at the same time, but not in eukaryotes. Why? Well, because eukaryotes have a nucleus that separates those processes, transcription and translation, in physical space. Transcription happens in the nucleus and to create a premature mRNA. That premature mRNA is chopped up, rearranged, capped, and poly A-tailed to create the mature mRNA. And then that is exported out of the nucleus and into the cytoplasm, where the ribosomes are waiting to link to it and create a polypeptide chain. And so transcription happens in the nucleus. Post-transcriptional modifications also happen in the nucleus. Translation happens in the cytoplasm, and any post-translational modifications will also happen in the cytoplasm. Prokaryotes do not have a nucleus, and so transcription and translation happen simultaneously. So here's our DNA molecule. An RNA polymerase is creating this mRNA from this RNA molecule, but while it's still working, ribosomes have already bound to the other end of the mature RNA and are making protein before the mRNA is even done being made. That is a process unique to prokaryotes because 1, they don't have a nucleus that separates transcription and translation in space, and 2, they don't do anything to their RNA. They don't do RNA processing. And so an mRNA, as it's being made, is as mature as it's going to get.